At the break, I was asked by several people about my comments about the aging debate, and this will be my only comment on it, and that is I understand that optimists greatly outlive pessimists. It's only 10 years ago this month when we published the first sequence of a free-living organism, that of Haemophilus influenzae, that took a genome project from 13 years down to 4 months. We can now do that same genome project in the order of 2 to 8 hours. So in the last decade, a large number of genomes have been added, most human pathogens, a couple of plants, several insects, and several mammals, including the human genome. Genomics at this stage of the thinking from a little over 10 years ago was by the end of this year, we might have between 3 and 5 genomes sequenced. It's on the order of several hundred. We just got a grant from the Gordon and Betty Moore Foundation to sequence 130 genomes this year as a side project from environmental organisms. So the rate of reading the genetic code has changed. But as we look what's out there, we've barely scratched the surface on what is available on this planet. Most people don't realize it because they're invisible, but microbes make up about a half of the Earth's biomass, whereas all animals only make up about 1/1000th of all the biomass. And maybe it's something that people in Oxford don't do very often, but if you ever make it to the sea and you swallow a mouthful of seawater, keep in mind that each milliliter has about 1 million bacteria and on the order of 10 million viruses. Less than 5,000 microbial species had been characterized as of 2 years ago, and so we decided to do something about it, and we started the Sorcerer II expedition, where we're, as with great oceanographic expeditions, trying to sample the ocean every 200 miles. We started in Bermuda for a test project, then moved up to Halifax, working down the U.S. East Coast, the Caribbean Sea, the Panama Canal, through to the Galapagos, then across the Pacific. We're in their process now of working our way across the Indian Ocean. It's very tough duty. We're doing this on a sailing vessel, in part to help excite young people about going into science. The experiments are incredibly simple. We just take seawater and we filter it, and we collect different size organisms on different filters, and then take their DNA back to our lab in Rockville, where we can sequence 100 million letters of the genetic code every 24 hours. And with doing this, we've made some amazing discoveries. For example, it was thought that the visual pigments that are in our eyes, there was only 1 or 2 organisms in the environment that had these same pigments. It turns out almost every species in the upper parts of the ocean and warm parts of the world have these same photoreceptors and use sunlight as the source of their energy and communication. From one site, from 1 barrel of seawater, we discovered 1.3 million new genes and as many as 50,000 new species. We've extended this to the air, now with a grant from the Sloan Foundation. We're measuring how many viruses and bacteria all of us are breathing in and out every day, particularly on airplanes or closed auditoriums. We filter through some simple apparatuses. We collect on the order of 1 billion microbes from just a day filtering on top of a building in New York City. And we're in the process of sequencing all that at the present time. Just on the data collection side, just where we are through the Galapagos, we're finding that almost every 200 miles we see tremendous diversity in the samples in the ocean. Just to try and get an assessment of what our gene repertoire was, we assembled all the data, including all of ours thus far from the expedition, which represents more than half of all the gene data on the planet, and it totaled around 29 million genes. And we tried to put these into gene families to see what these discoveries are. We just discovering new members of known families, or are we discovering new families? And it turns out we have about 50,000 major gene families. But every new sample we take in the environment adds in a linear fashion to these new families. So we're at the earliest stages of discovery about basic genes, components, and life on this planet. When we look at the so-called evolutionary tree, We're up in the upper right-hand corner with the animals. Of those roughly 29 million genes, we only have around 24,000 in our genome. And if you take all animals together, we probably share less than 30,000 and probably maybe a dozen or more thousand different gene families.